ahead and, all right, sorry, thanks, Rosie.
Perfect. Thanks, Thomas. And thanks, Evie. All right. So now looking at the training agenda, just wanted to give you guys an overview of what to expect for today. So going to do a quick overview and introduction. Thomas is going to lead that. And then we'll get into some harmful algal bloom phytoplankton identification overview. Thomas will do the introductions when we get there. There will be some time for expert discussion and Q&A. So feel free, if you have questions as they pop up, to enter them into the chat as they're coming to you, but we will address all of them when we get to the Q&A, just so that we have enough time to kind of work through those different presentations. We will do a short bio break for about 10 minutes, and then I will be going into our phytoplankton sampling techniques. So these are phytoplankton sampling techniques that we do at the Research Reserve, very similar to what's done by the group down in Southeast with Seator, Sitka Tribe, and also the Phytoplankton Monitoring Network. I'll go through some different resources that we have as well, and then we'll do a training wrap up before we adjourn, and we're aiming for around 10:45 today. All right, great. Thanks, Thomas. So just a little bit of background before we get into the introduction on who we are. You know, I mentioned that this training is being hosted by the Kachemak Bay National Estuarine Research Reserve and the Alaska Harmful Algal Bloom Network. So the Kachemak Bay National Estuarine Research Reserve, this is a partnership between both NOAA and the University of Alaska Anchorage, the Alaska Center for Conservation Science. And those two groups make up us at the reserve. We're located in Homer, Alaska, and we are the only research reserve located in the state of Alaska. Our phytoplankton and harmful algal bloom monitoring program has been going on for the past 16 years. And our goal with this program is to really be able to continue monitoring for harmful algal blooms in South Central Alaska and take some of the different things that we've learned and share them with folks around the state who are beginning to do monitoring themselves. Then the Alaska Harmful Algal Bloom Network, this is located within AUS, the Alaska Ocean Observing System hosts it. And AHAB was formed in 2017 with this overarching goal of taking a statewide approach to monitoring, research, response, and education and outreach has been the overarching goal there with AHAB. You know, prior to this, and especially after the warm water blob event that we saw happen in 2015, there was just a lot of concerns about harmful algal blooms. This was growing in the state of Alaska, and new monitoring programs were starting to pop up around the state. And it really felt like it was time to bring us together to try to take a unified approach, which started the Alaska Harmful Algal Bloom Network. And then just this past year, we're able to welcome Thomas on as an official coordinator in the role, which has been great. So that's a little bit of a background on these two groups. And with that, Thomas, I'll throw it back to you to get started with the introductions.
Perfect, excellent. Thank you. Thanks for that, Rosie. Yeah, so let's just dive right into it so that we give plenty of time to the panel to talk to us about harmful algal bloom identification. So I know most of you have seen this before, but just as a reminder, harmful algal blooms, they occur when colonies of algae grow out of control and produce toxic or harmful effects that usually impact people, fish, shellfish, marine mammals, and birds. And these can cause illness in humans as well as in other organisms as well. But it's important to remember also that algal blooms are not always harmful. In fact, this picture here is of a non-harmful algal bloom, and this is just the spring bloom in the Bering Sea, which is an integral part of the ecosystem, really. And even within harmful, the term harmful, there's really two categories of harm. There could be non-toxic effects where just the sheer number of cells in the water can create anoxic or hypoxic conditions that removes all the oxygen. That could be very detrimental. A lot of cells in the water can also clog fish gills and lead to suffocation. And then there's also socioeconomic impacts sometimes in some regions due to the discoloration or turbidity of the water. And this is places that have high tourism like Florida that may see economic loss because of a red tide or something like that. And then within the toxic effects, this happens when these cells are not only really abundant, but also produce toxins. And so the more cells there are, the more toxins there are usually, which is why we're trying to be aware of these bloom conditions. these impact humans and wildlife. And the effects that are felt by humans and wildlife depend on the toxin itself that's being produced, and therefore on what organism is in the bloom condition. So that's why it's important to identify what HAB species we're talking about. And in this case, we're talking about species kind of in quotes, because really we're talking about groups of organisms that are more at the genus level. And it's pretty hard sometimes to identify down to the species level, but we're going to use the term species just for simplicity. But know that when we talk about things like Alexandrium, Dinophyces, this is actually a genus, not a species. But like I said, phytoplanktons are an important part of Alaska's marine ecosystem, and identifying what is blooming can tell us whether or not it's a harmful bloom or not. But also within a harmful bloom, it could tell us whether or not, if there's that specific species in the water, what we might expect, what kind of warning we might want to give to people and to fishers and to harvesters, etc. The bottom graph here is from Kachemak Bay National Ecological Research Reserve, and this is just which of these 3 HAB organisms were present throughout the year in 2020. And so you see that all 3 of them, which we'll talk about today, were present at some portions of the year, but there were no blooms of any of these actually happened in Kachemak Bay in 2020, but they were present to some level, just not under bloom conditions. And then the other important part of seeing when these organisms are in the water is if we can also get additional data at the same time, just temperature and salinity, we can start understanding what are the dynamics, what are the conditions that produce a bloom event. So obviously we need the organism there, but then what other sets of temperature, salinity conditions, or other environmental conditions switch from just the presence to a bloom condition. So here are the ones that we'll touch on today. I won't go in too much depth right now because we'll go through each one, but just to give you an overview. So Alexandrium is probably the one that we talk about the most in Alaska. It's what leads to paralytic shellfish poisoning. And so, it can create a lot of health effects. And this was the organism responsible for the mortality in July 2020 in Alaska. Pseudonychia is also talked about quite a bit, mostly for its impacts on wildlife. It produces amnesic shellfish poisoning and that kind of drunken behavior that is seen in marine mammals sometimes, especially in California. We'll also talk about Dinophyces, which hasn't yet been, the effects of it haven't been yet reported in Alaska, but we do see the organism in the water. And then Karenia, which we've seen a few blooms in Kachemak Bay. They haven't been harmful so much, but they are, a Karenia bloom was detected, if you've heard, in October in Russia on the Kamchatka coast. That created a lot of organisms, fatality of crabs and invertebrates and fish, and a lot of impacts on people's respiratory systems as well during this kind of red tide event that happened there last fall. So not impossible to see it having the same kind of effects in Alaska at some point as well. So that's it. We'll go through each, how to look at each one individually in a minute here. And the people that will help us do that are these experts here on our expert panel. So experts, feel free to amend whatever I say here, but just to give a quick overview, you guys have all heard from Rose Masui already. She's the Harmful Species Program Coordinator for the Kachemak Bay National Estuarine Research Reserve and has looked at phytoplankton for a long time. So a great expert. She's also leading the efforts to produce the K-Bay NER phytoplankton guide, which is a great resource. Dean Stockwell, UAF Associate Professor, great expert on phytoplankton as well. So his expertise is going to be really helpful. And then I'll just mention Karie Holtermann because she's also based in Alaska, in Unalaska actually. She's the lab manager of the the City of Unalaska Water and Wastewater Laboratory. And then we've got 2 great experts from Woods Hole Oceanographic Institution, Dave Kulis and Evie Fachon. Evie leads a lot of the cruise, or is on a lot of the cruises that are going up in the Arctic looking for Alexandrium and other harmful algal bloom species. And then Kate Hubbard from the Florida Fish and Wildlife Conservation Commission. She's a great expert on Pseudonychia and Karenia, does a lot of genetics work on those organisms, and has projects in collaboration with people in Alaska. So thank you to these experts for coming and talking to us, and I will turn it over. And so just a quick note, I will be running the slides, and so I'll try to run these as smoothly as possible, but please mention when you want me to move forward if I'm not moving fast enough for you. So let's start off. And sorry, I just saw that a couple questions about whether the slides will be provided, and yes, we will be providing these slides, and the recording as well will be made public. So we'll start off with HAB phytoplankton ID, and I think Evie is going to start us off with Alexandrium. Or is that right? Is it Evie? Yeah.
Yes.
Yes.
Awesome. Thank you.
Yeah.
Great. So Alexandrium, as Thomas just mentioned, is a dinoflagellate that causes paralytic shellfish poisoning. And so it belongs to a group of algae known as dinoflagellates, and these have these little whip-like structures called flagella that they use to propel themselves through the water. Alexandrium specifically is generally a rounded cell. A big characteristic of it is the reddish brown or golden color, as you can see in this image on the left. It's also what is known as a thecate algae, which means that it has these plates or armor around the cell. And these can be a little difficult to see with regular light microscopy, but it is something to keep an eye out for when you're looking at your samples. And then also Alexandrium has this deep waistline groove known as a cingulum. I've included some arrows here to point that out. And Thomas mentioned the other day that this makes the cell look like sort of a squashed hamburger, which is a description I've never heard before, but that I really like. Next slide, please. So as Alexandrium grow and divide, they can stick together in pairs or chains. I've seen chains of up to 8 cells long, but I've also seen plenty of samples that just have single cells. So these are some examples of what that might look like. I've generally found that a lot of the Alexandrium we've seen in the Chukchi Sea and in the Northern Bering tend to be the single cells or the doublets, while down in Southeast, it seems like you guys have more of these chain style Alexandrium that look like sort of strings of beads. Next slide, please. So if you don't mind playing this video, we can have a look at what these cells actually look like in culture. Depending on whether you're looking at live or preserved samples, you might see some guys like this swimming around. And the characteristics that you can discern will depend on the power of your microscope. So at the lowest magnification, you might just see these sort of like golden balls tumbling around. But if you're able to zoom in more, you can get an idea of some of the structures that we looked at before, such as that waistline groove or even the flagella in some cases, although those can be kind of hard to find. This cell right here that you're watching sort of roll around is maybe not the healthiest cell. It looks like it's not doing super well, but it's a good way to sort of see some different angles of what the cells look like. So I provided Thomas with a link to this video, and if you want to go back and watch it again, you can get a better idea of what you might be able to look at in some of your live specimens. Next slide. And then really quickly, this is a chain cell swimming around. So they kind of do this cool corkscrewing motion as they move through the water. Thanks. Next. So there are definitely several other organisms that you can see in your field samples that can be really confusing and can look a lot like Alexandrium. I've included just a couple of those here. So one thing that you'll notice, on the left, we have our Alexandrium cell. It's golden, it's round. On the right, we've got a couple of these more confounding species. So Scripsiella, the one on the top, has this sort of pointed vika to it. It sort of looks like a party hat, and you would never expect to see something like this on an Alexandrium. Heterocapsa below also has kind of like that pointed shape. And so again, if you're seeing structures like that, even if it's the right color and the right size, it's probably not Alexandrium. And then the one on the right is even trickier. And so if you don't mind advancing one more time, Thomas. It is a chain. It's that dark pigmented color, maybe a little darker than we might expect to see for Alexandrium. But if you look really close at the bottom, you'll see that it has these little spikes on it. And so this tells us that that's actually Paradiniella, which is another chain-forming dinoflagellate that you can find in the Arctic and can look a lot like Alexandrium. So just keep an eye out for these points and spikes in any sort of structure that seems abnormal, because those are all good signs that maybe you have something else swimming around in your sample. Next. And then finally, I just wanted to mention Alexandrium has this really cool life cycle where in the winter, when it's cold and dark and maybe not a great time to be an algae swimming around in the water, they actually form what's known as a cyst stage, which is kind of like a little seed that settles down into the bottom, and it doesn't do anything at all. It just waits for the proper light and nutrient and warmth cues to tell it to bloom again. And the reason that I'm mentioning this is that we've actually sometimes seen these cysts in our water samples. And so what they look like are these sort of pill-shaped oblong cells, and they can be on their own or in clumps. They sometimes have a little mucus around them, and we see them generally in like the late summer. So just keep an eye out when you're looking at your water samples, because there might be some of these in there as well. And with that, I'll pass it on to Karie and Kate, who are going to talk about Pseudonychia.
Great. So Pseudonychia, switching gears a little bit, is a diatom, and it forms needle-like chains, although it can occasionally occur in single cells, as you can see in the image shown here. And if you go ahead and press play, Pseudonychia doesn't swim, but it does glide. This is it gliding on a microscope slide. So you may see it moving around if you're looking at a live sample. And it's unlikely to be, again, swimming in the water, but it may be able to glide across substrates. And sometimes the cells can even glide across each other if they are concentrated in a thin layer in the water. The cells have pointed tips, and there are different types of chain formation, so we'll go over that a little bit. That tends to vary from species to species. And one of the big things with Pseudonychia in the Arctic and across the world is that there are a lot of different species. Some of them we know, and some of them are likely yet to be identified and fully taxonomically resolved. And so with Pseudonychia, only some species have been shown to be toxic. And then we also know that that toxin production can be triggered by different factors. And these can include environmental factors related to nutrients or temperature, but they also can be triggered by grazing activity in some cases for some species. And some of those studies have been done specifically with polar Pseudonychia species. So I thought it was helpful to mention that. The species cannot be differentiated by light microscopy, so it's good to point that out. Next slide. So I'm going to hand off to Karie.
I'm not sure if you can see me here. I am just going to mention that there are several types of Pseudonychia that I see here in Unalaska, and this is likely in other places as well. And as Kate says, you can't really differentiate under the microscope, under a light microscope. You can under an electron microscope. And then she has molecular techniques that she uses that she's able to identify to species. But the two types of cells that I am seeing here in Unalaska are a very large cell that usually forms in chains, and it's greater than 3 microns, and then a very small cell that's less than 3 microns. And sometimes they're in chains and sometimes they're single cells. And I see these cells throughout the year, but there's 2 main times when they form blooms. And the only one that I see that forms blooms is the larger cell. And that's in the summertime and also in the late fall. And I just categorize them as the larger Seriata type cell and the smaller pseudodeli type cell. And that's the 2 categories that I put them in.
And if we look on the left-hand side at those large type cells, there are multiple species that can look just like that top one. And several of those species are known toxin producers. And if you look at the top versus the bottom image, you'll notice that the chains actually look different. So you can almost draw a straight line across one side of the top image. And then you can see the different curves of the cells as they're connecting. And on the bottom, you see more of a step-like chain where they're sort of blocked on top of each other. And so those are both characteristics of different species. And again, there are multiple species that fall within each of those different groups. And we use widths to try to discriminate between Pseudonychia, and there are some that fall right at that 3-micron size. And so, you know, those are kind of a pain for figuring out whether they're large or small, but generally we tend to lump them more with the larger ones. And then if we go to the next, this is just to give you a sense now, these are all on the same scale to give you a sense of some of the diversity, again, moving from large to small. These were all observed during a single cruise from the Bering Sea to the Barrow Canyon. And so it helps, I think, to see that in perspective. Again, there's also a lot of different chain types here. And these are all different types of Pseudonychia, and you know, I can't tell you what species they are. So it's just good to point that out. And again, these are good references just to kind of give you a sense for what they look like. And as we move to the next slide. Oh, good. There's some better images. I put in some artistic renderings that I drew in PowerPoint, but these are much better. These are from Karie. So there's some species that can tend to look a lot like Pseudonychia, but typically are not toxic and may or may not be closely related. And Karie, I don't know if you want to go through these since you popped them in, but the things to look for, you may see some of these in single cells, and so it may be difficult to tell whether or not it's Pseudonychia or one of these other things. When they form chains, then you can see that there's these different types of chains that are formed, and some of them may end up stacking, like Fragilariopsis or Fragilaria. There's another one in the middle, which is Leptocylindrus. And that one, those are cylinders, but those can get really, really small and sometimes look very similar to some of the smaller Pseudonychia types. And then there are some of the single-celled ones, again, that can be really confusing. Basilaria is a really fun one. It's too bad we don't have a video to show, but that one is quite an active mover. Those cells can slide against each other, and so that would be something to look for that Pseudonychia doesn't do. And then Rhizoselenia is another one that can sometimes, there's my dog, look like Pseudonychia. But if you start to look at the tips of the cells and how they're connecting in those chains, you can get a sense, and Rhizoselenia has some different features that differentiate it.
Karie, do you want to say anything else about this slide since you put in some of these pictures?
Just that Pseudonychia usually will be in chains, and that the single-celled pinnate diatoms are usually, well, they're never in chains, and they're always single cells moving pretty quickly. And Basilaria actually is a freshwater species, but also found in brackish water. And it is found in Unalaska quite often. And one of the cells that we see a lot is, there's a lot of Pinnellarias here too, which are also a freshwater species, but it's found here in the salt water. But usually Pseudonychia will be in chains.
Great, awesome. All right. Are we good with Pseudonychia? Yeah. All right, Dave.
Okay, I'm unmuted.
Yeah.
Can you hear me?
Yep.
Okay. Hello, Alaska. I'm I'm Dave Kulis, and I'm here to talk to you a little bit about dinophysis. So, and our IS department just notified us that we're experiencing internet connectivity issues. So if I glitch out all of a sudden, that's the reason why. It's beyond me. So dinophysis, it produces nasty toxins, okadaic acid and dinophysis toxins that result in the syndrome diuretic shellfish poisoning. As the syndrome name implies, gastrointestinal symptoms may result as soon as 30 minutes after consumption of shellfish, and they may last for up to 3 days. So it's something you just don't want to tangle with. The toxicity of dinophysis cells are very highly variable. You can have toxicity with as little as just 1,000 cells per liter, and those can contaminate shellfish, or it may take over 1 million cells per liter to contaminate shellfish. And this is dependent on the species and also upon the geographic location. For instance, Diacuminata in Europe are very toxic and on a per cell basis, but here in the Northeast United States, these cells can be mildly toxic, where it takes over 1 million cells per liter, a sustained population, to be there for maybe a week or two before you see an uptick in toxicity. Next slide, please. Next slide, Thomas. Thank you. So the nice thing about dinophysis is that they're relatively easy to identify. The 3 species that you find in Alaskan waters, at least that have been noted so far, are Tripos, Norvegica, and Acuminata. And the ones that I have highlighted with the red circles with the line through them are the toxic species. So in addition to those, there's Caudata, Fortii, and Acuta that are known to produce toxins. And these cells could be confused with Phallochroma, and there's another genus also that has cells with a similar morphology. And we'll sort of, we'll try to address that as we go on here. Next slide, please. Okay, I gotta get my notes on the right page. Bear with me. Get to my other screen. Okay. So there are a couple of key taxonomic features that are seen on dinophysis cells that aren't seen on the other cells that you've been introduced to today. One is the singular list, which, Thomas, maybe you can point with your mouse to the singular list, and then the sulcal list. So the singular lists are at the top of the cell, and the sulcal lists are on the side of the cell. Right, exactly. So we don't really need to go talk about singular and sulcal. We can call it the crown and the wing. And those are distinctive features of dinophysis. And this is on the right-hand panel. We're showing you a dinophysis norvegica cell. And dinophysis is really interesting in that it's mixotropic. The 2 cells or 2 species that you were introduced to before, Alexandrium and Pseudonychia, are phototropic. They photosynthesize to produce their food. Whereas dinophysis actually eats other organisms and also photosynthesizes. And it feeds through a peduncle or a feeding straw, and it captures these little ciliates in panels C and D, and those are called mesodynium cells. And the mesodynium actually eat the little cell in panel E, which is a cryptophyte, teleolax. So it's a 3-tiered system where the teleolax feed the mesodynium, and the mesodynium feed the dinophysis. And so if we go to the next slide, please, we can see, actually go back one. This is the next slide. Okay, so this is an aerial view. Oh, skip down. Next one, please.
Sorry, there's a bit of a delay. Is this the one that you want with the different?
Sure, we can start with this one. Thanks, Thomas. Yep. So on the left-hand panel is a Dinophysis acuminata, and you can see the little peduncle that is going in from the Dinophysis cell into the ciliate. And basically, it's going to suck the contents of that ciliate through that peduncle or feeding straw so that you can get its mixotrophic nutrition. The other interesting thing to note here is that Dinophysis acuminata has a very rounded bottom, whereas Dinophysis norvegica has a very pointy bottom, or more of a pointy bottom. So those two species can be, I think, somewhat easily distinguished from one another. And they certainly can be distinguished from Dinophysis tripos, which has these processes coming off of the posterior part of the cell. So, and this is a dividing cell of Dinophysis tripos. So it's fully divided. The cells are still stuck together, but soon they will break apart, and these two cells will again divide through asexual division and form two more cells. When I was looking through some samples that came from the Alaskan waters recently, I was finding this rather large Dinophysis cell, which I thought was a Dinophysis cell. And I thought initially it may be Dinophysis acuminata, but it just was too big, too broad. And in trying to key it out, I realized that it was an entirely different genus called Phallochroma. And this is Phallochroma rotundatum. And you can see that it's much broader in appearance and maybe a little bit, well, I don't think it's more squat. It's much more broad than Dinophysis acuminata. So anyways, bear that in mind. You might want to consult a guide like Identifying Marine Phytoplankton by Tomas. If you can get a copy of this, it's a wonderful resource to have, and I highly recommend having that at your disposal when you're checking out samples. Next slide, please, Thomas. Okay, so on the left-hand panel is a aerial view of a mesodinium bloom. And you can see that it's a very patchy phenomenon and also a very dense bloom former. These are just huge accumulations of these little ciliates that form in the water. And you would expect that if you have high concentrations of these ciliates in the water, that it could be followed or coincide with a Dinophysis bloom. So if you are seeing dense blooms of the ciliate, this pink water, you might want to try and grab some water samples and see whether or not Dinophysis might be co-occurring with the ciliate. And if we can snap on this video, there's a Dinophysis cell with a peduncle in the left-hand corner, upper left-hand corner. And you'll see the peduncle right there. And so this was a video that was taken when we supplied these cells with a whole bunch of food, and they're very antsy, and they're all swimming around with their peduncles out, looking for ciliate prey to try and feed on. And there must be some ciliates in this little clump of food, because if this video was to go on further, you would see that more and more Dinophysis are coming over here, attempting to graze on the cells. Okay, we can go on to the next slide, please. And this is just kind of a cute little video that I found where even Dinophysis, even phytoplankton cells can be aggressive. And look at this guy right here. He's just bossing this other Dinophysis cell around and pushing him away. And, you know, it's like, you just can't escape it, you know? There's bullying happening everywhere in culture. But that's what I have to say about Dinophysis, and I'm going to pass the baton over to Kate, I guess, or to Karie.
Thanks, Dave. Yeah, I can't remember. Is it Kate or Karie? But Kate, okay. Go ahead. Oh, do you need to be unmuted? Okay, yeah.
I'm unmuted now. And the mailman has left, hopefully not to return. So Karenia mikimotoi, or just Karenia, is an unarmored dinoflagellate. So unlike Alexandrium, which has that sort of protective shell, Karenia does not. And so it pops a little bit easier. And it also can swim in a little bit of a different way. Unfortunately, I don't have a video of it swimming, but you can almost imagine them like pancakes, and they can move like this with their different lobes. They tend to be reddish or brown, and they've been referred to, especially in some of the Alaskan blooms, as being amber. And when they discolor water, the Alaskan bloom that happened in 2013 was referred to as a beer tide because of that color. And when you see a water sample that's just full of Karenia mikimotoi, it really is this sort of dark amber or brown color. These tend to be oblong cells, but they can also be round. And they do tend to be longer as opposed to wider. And there's a round on the top, and then you see that lobed pattern on the bottom. And in the image on the right, you see cells in a few different orientations. And so they can look different depending on which view you have. And you see one even on the lower end that looks just kind of like nothing really. Sometimes if you flick your slide, you can get them to reorient, and then you can see some of the features that are helpful again for telling Karenia apart. Next slide, please. So these are some of the different Karenia species that occur. So far, only Karenia mikimotoi has been observed in Alaska. It does have an oblong nucleus. It can extend across the epitheca and hypotheca, so that's the top of the cell and the bottom of the cell. But typically it's in the hypotheca, so it's in the bottom. And even though it looks like it's on the right side, remember this is sort of a mirror of what's actually being seen in the cell. And so it's usually on that same side, but it's actually on the left side of the cell. But for the sake of looking at it under the microscope, it's going to show up on the right side. And the cingulum, if you notice, so that was the groove that we've seen in some of the other species, that the dinoflagellates. If you look at that and trace it across the cell for mikimotoi, it's displaced. And so that's another feature that we can use to differentiate that from some of the other species. And if you look at those other species, those are all species that we see in the Gulf of Mexico and other parts of the US, with the exception of Karenia brevis, which is only in the Gulf of Mexico. You can see that nucleus changes position. Next slide, please. So you can't always see the nucleus, so that's just something to keep in mind. There are two flagella, one that goes along that groove, and then one also that comes out of the hypotheca, or the bottom of the cell. And it is, as I mentioned at the beginning, a swimmer, and those lobes will help with its movement as it moves around. It can form very massive blooms, over 10 million cells per liter. I think some of the highest blooms I've seen reported approach 70 million cells per liter. And as that happens, the cells lose shape, and they also lose those key features, and it can make it really difficult to identify. And you can see an image from a sample this is actually from a main bloom, but this is around 35 million cells per liter. And you really can't see any of the identifying features. And so then you look to some other things like the color of the water. And for this one, we actually did have to do genetic ID to confirm that. But it does give you a sense. And then this is also, it's great to take these sorts of shots with your phone if you can, because you never know, that might be, something that you can pass on and keep. But in a dense bloom, again, these can be really hard to identify. Next slide, please. I just wanted to show this because it does show some of the morphological variability within just this one species. And so you can see that there are different cell types, different cell stages. Evie mentioned at the beginning that Alexandrium can form that cyst stage that is sometimes observed in water samples. And what sometimes we see for Karenia when blooms start to become unhappy, and I kind of showed this a little bit in the last image too, we do see that cells start to look more rounded. And there's been some question as to whether or not they can form some sort of resting or potentially sexual cysts, and whether those cysts could then germinate and potentially go back into a regular vegetative cell. And so this just gives you a sense that, you know, if you are seeing some of these round cells either after a Karenia bloom or as part of a Karenia bloom, those might in fact be Karenia cells. And Mikimotoi is not known to produce toxins. Well, most of the toxins that it produces are thought to be more ichthyotoxic, so more harmful to benthic species and also to fish. However, you can imagine with those high density blooms when they form, there's other impacts just from the bloom decomposing, and so there may be mortality associated with them. I think that's it.
Excellent, thank you. That was a great overview of those 4 HABs, and so, but you'll see other things under the microscope, and so Rosie's going to show us a few other ones as well that we might see.
Yeah, great. Thanks, Thomas. If you want to hop to the next slide. So I'm going to do this a little bit differently and kind of tell a little bit of short stories about these ones that we can see and have seen in Kachemak Bay. So the photo on the right-hand side is me looking under the microscope and seeing a Catosphorus bloom. And so what we had been happening here in Kachemak Bay, this was in the early 2011, what all of a sudden we were hearing from some folks is that there were some issues in the fishing hole in Homer. So if you've ever been to Homer, there's a large peninsula called the Spit, and on the Spit, there's this area that's a fishing hole, and it's this great spot, and it is where the local Department of Fish and Game stocks salmon. And this is a great resource. So if you want to go fishing, but don't want to pay to go out in a charter, and the weather's bad, you can go right to the fishing hole. So the Department of Fish and Game brings juvenile salmon from Anchorage, trucks them down to Homer, and they pump them into net pens in the fishing hole. And what was happening is all of a sudden these fish were dying, and they were not sure why. And that's really a nightmare, right? Because you think about how much money goes in to raising just one of those little juvenile salmon. It's a good size price tag. And also this had implications because the whole goal with stocking the fishing hole is that those salmon will go out to sea, grow up, get bigger, and then come back and then can be harvested in a few years. So there was concerns about what's going on. And when they went out and collected a phytoplankton sample because they were like, okay, what's going on in the water? Let's at least take a picture, see what's happening. This is what they were seeing. They were seeing a large-scale Catosphorus bloom. So Catosphorus is a diatom. This is, diatoms, like was previously mentioned by Kate and Karie, the Pseudonychia is also a diatom. Well, Catosphorus is a different species or genus of diatoms. There are over 400 Catosphorus species worldwide, and it's a really, really common species to see in our samples in Kachemak Bay. Thomas, if you want to pop to the next slide. So there's Catosphorus concavicornis and Catosphorus convolutus. Just another little thought out there too is when I've been doing my phytoplankton training and learning taxonomy and trying to get some of these different names down, what I've been told over the years is that if the person on the other line of the phone can understand what you're trying to say, then you pronounced it right. So to not worry too much. So if folks are getting started on monitoring and looking at things under the microscope and they're feeling a little bit concerned, like, oh gosh, am I just not pronouncing this right? Don't worry about it. It's okay. It'll come through. So this species specifically can potentially be a fish killer. But it's a specific situation that can happen. And so the fish really need to be penned. And so what can happen and what was happening in this situation is that there was a large scale Catosphorus bloom. And so since these juvenile salmon weren't able to dive underneath this bloom, the Catosphorus was getting caught in their gills, as you can see from this image right here. And Thomas, actually, will you go to the next slide too? Awesome. See how big those and intense those spines are. And that's really characteristic of Catastrophes, is these big spines that can be attached to the different cells. And so what was happening is that these spines can lacerate the delicate gills of small fish or cause an irritation. And when they cause that irritation, a fish kind of reacts in a way that we do when we're sick. So they release a ton of mucus. The goal is trying to flush these things out, but they're so clogged with catastrophes that they actually end up dying. And that's what ended up happening in this case. They were suffocating to death. So this can be avoided. I don't want folks to think, oh my gosh, I saw catastrophes in the waters, we're expecting to see a big scale die off. That's not necessarily what's going to happen. The situation kind of has to be this perfect storm and for this to happen and lead to fish kills. And so areas like hatcheries, and where they're stocking salmon potentially in Alaska could be vulnerable. The salmon weren't able to dive underneath this Catastrophes bloom, which led to this die-off event. What the local Department of Fish and Game does now is they do a phytoplankton tow before they stock salmon. They're doing the phytoplankton tows while they're in the process as well, while the salmon are in there, to make sure that a large-scale catastrophe bloom isn't happening again. And if it is, all they need to do is drop the nets in those pens, and then the salmon can dive underneath. The fishing hole after that incident also had to be dredged out to give a little bit more room as well. But this is just something to keep in mind. So this, compared to some of the other species that we were hearing about, isn't producing toxins. Catastrophes is also super important in the marine food web, and it really doesn't do have a lot of apparent harm to animals that are eating it. But it's really that situation with fish and net pans that can cause some problems. There's also situations where those spines, when they are causing those lacerations on fish gills, where they can also introduce bacteria into the bloodstream of fish as well. So that's another way that they can cause some problems. All right, and these are pretty dramatic cells to see under the slide. They can be quite large, and what we're looking at right here is looking at not the, we're seeing it at the girdle view, so it looks kind of rectangular and long, whereas if you're seeing it kind of like top down, the valve view cells are round. So they can look a little bit different, but I would say one thing to do also is to make sure to take photos, so I think that's something else when we get into the sampling section of this, is we can talk about some tips about, if you are sampling phytoplankton, how to collect some photos, even just using your iPhone and the microscope. All right, Thomas, if you want to go to the next slide. So then this is a recent one that I wanted to mention as well. So another story of something happening in Kachemak Bay that just happened. Thomas, if you'd hop to the next slide. So this is a photo that was taken in May of this year by a pilot flying over Kachemak Bay. They were actually doing some aerial herring surveys with the Department of Fish and Game, went out in the water and saw this intense view, saying the water's red. And so a lot of folks on here have probably heard the term red tide. This especially, I'm in the lower 48. Kate, where you're at in Florida, this is a big concern. The term red tide has a lot of negative and intense connotations associated with it. And so immediately people were pretty concerned. I heard about it early on from some of our community monitors in Kachemak Bay, but then when it got posted on Facebook, there was a lot of concerns. People were saying, is this PSP? Is this, are we having an attack by aliens? Somebody wrote it, it's Facebook, so people kind of went crazy with it. We were able to go out and get a sample, and Thomas, if you go to the next slide, this is what we were seeing underneath the microscope. And Dave kind of set this up for me pretty perfectly. So what we were seeing was this marine ciliate. And so ciliates, these are these planktonic guys that have cilia. And so you can kind of see those little hairs around this guy. Yep, Thomas is pointing to them. And those are used to help them either move around in the water or to go and grab food. And this species has a pigment in it that's red. And then it was doing, like Dave kind of walked us through that scenario, was eating a cryptophyte that also had a red pigment. And it kind of led to this intense bloom, as we call it. So this large scale amount of an exponential growth of this marine ciliate, which colored the water. But it's not harmful, although it's super alarming, right? You know, you want, ideally, it would be great if these harmful algal bloom species that we're paying attention to colored the water, because then we could go outside and be like, oh, water's red, might not want to eat some wild shellfish today. You know, that would be great. But that's really not the case, you know, and it also can lead to a lot of misinformation. So the term red tide, I kind of always am, dread it in Alaska sometimes, because it comes with some confusion in our state, because often when we're seeing things like Alexandrium, which a lot of folks associate with the term red tide, it's not coloring the water. You know, Dave showed those great photos of Dinophysis where it can color the water. And so far in our sampling efforts for the past 16 years, we haven't seen these harmful species outside of Carnea mikimotoi really color the water intensely. And so that's just something to keep in mind. So this is an example of a bloom of a marine ciliate that's a plankton that was really alarming for a lot of folks, but it wasn't harmful. And so other things to keep in mind is that folks like oyster farmers might then see some time periods where their oysters actually, the meat actually turns red, which to a lot of folks can be pretty alarming. You know, so if you're going to harvest wild shellfish, oh my gosh, these mussels are redder than usual, or the clams are red. It's because of what they're eating. That doesn't necessarily mean that those shellfish are toxic. You know, especially in the state of Alaska, if you're going out and you're buying something that's commercially harvested, it's regulated by DEC. And so that's been tested, so you're safe to consume that. So there is a lot of misinformation out there about these, but just wanted to highlight this. So if you are seeing this big water change, reach out and connect with us, connect with different experts. We can help you figure out what it is, because it might not be toxic. All right, I think on to the next one, Thomas. Yeah, expert
Great.
discussion.
Yeah. Thank you, everybody. That was fantastic. I actually learned quite a bit as well. So hopefully everybody got a lot of good information out of that. And so if you have any questions, feel free to start putting them into the chat. We're running a little bit behind, but I think Rosie mentioned that the sampling side of things won't take a full hour. So experts, I know we didn't talk about this specifically, but if you're willing to stay on for a few extra minutes past 10 a.m. Alaska time, we can go on for a few extra minutes here. And if anybody has any questions yet, please put them in the chat. But maybe to start us off, I'll see if Dean Stockwell has anything to add on his experience about HABs and HAB species in Alaska. I don't know if Dean, I think Dean, you're unmuted if you want it.
Yep, I'm unmuted. I'm here.
Hi, great.
So I think the most important thing that comes to mind is eyes on the water. Local people are the ones that see these events taking place, whether it's a red water event, a white water event, any types of those things get reported. Rosie mentioned the Mesodynium bloom. We had one several years ago off of St. Paul in the Bering Sea. These are common events that take place, but people have to reach out and say, we see this thing that's different. Pink oyster meat, yeah, is this toxic? And then people like Rosie, like K-Bay, like Seator, or any of the groups that are monitoring can say, yes, we know about this. It's not necessarily a harmful bloom. Down in Southeast, it's very common to get pink water, orange water, and various shades of red, all caused by Noctiluca, which is another type of dinoflagellate. And these are very common events. They don't have toxins themselves, but massive blooms, as David mentioned, can deplete the water of oxygen, and you can get small fish kills that way. And those things are concerned. Catherine deals with a species that's very difficult to identify, even for the experts through the microscope. And they take a lot of prodding and scanning electron microscopes and DNA work to do their work. But we know those are very common species in Alaskan waters. In fact, some of the state DEC information on domoic acid focuses primarily down there in Homer. Some of the highest concentrations measured by the state are in Homer. And so these are things to be aware of as a community sampler. And if you see something in large numbers, Catostris is a great food organism for most zooplankton. Yes, the big species can come in there and irritate the gills, and they can have problems on young fish that can't avoid it. But if we don't have those types of blooms reported, you know, we're kind of up in the air. So if you see something in large numbers, get a photograph of it, or even get a water sample of it, and maybe you can get it to Rosie or some other group in the state that'd be willing to look at it. And I guess eyes on the water, it's really important. And if you think something looks suspicious, if you noticed fish kills or marine mammals acting oddly, and you have a sample of water, you know, get a picture of it, just like this Catostris picture.
That's great. Yeah, thanks, Dean. Quick follow-up on that. If somebody doesn't have access to a microscope, but they see something in the water, is it good enough to just take an empty water bottle and just fill it up with seawater and then contact somebody to try to get it to them? Or is it more involved than that to
fill it up with seawater, put it in your refrigerator until you can contact somebody and try to do a next day shipment or whatever. We've gotten materials from off Kodiak. I've gotten the Mesodynium material from off St. Paul. I had discolored water off of Nome 2 years ago. It ended up being a massive pollen, a birch pollen bloom, but it turned the water gray. I got that material. And so, yeah, put it in a water bottle and get it to somebody that can analyze it. And, you know, probably Tom is your contact, call him. But I know the LEO network here in the state, Mike Brubaker will contact me if something's going on. So, yeah, reach out. There are people there that will help.
Perfect. Yeah, that's excellent. Thanks, Dean. Appreciate that. So kind of going off of what Dean was saying, and also a question from the chat, can somebody touch on how we quantify the word bloom and how many cells is considered a bloom, which I'm sure will differ depending on what's the organism that we're talking about. But it seems to be a little bit of a qualitative, subjective. There's just a lot of them in a sample. But is there any either quantity or is it just because if it's the dominant organism in a water sample that we would call it a bloom? Who wants to jump in on that?
Maybe I could try.
Yeah. Because
we have a lot of different operational thresholds that we set for blooms, and you can look in the literature and that, you know, there may be thresholds set there, but we work with so many different species and sometimes have things that we've never seen before. And so we often will set those thresholds ourselves based on, again, if we see something, looking at samples regularly. And for some species that we see more routinely, we may even have two thresholds. One that is something that's more biomass related. So once cells reach a certain threshold, they may start to discolor water. You may see them, again, at a high level of abundance. But for some of the HABs that can impact human health, there may be a lower threshold that is important to be aware of, and that threshold again can vary across all of those different species. But for Alexandrium, you know, some of the Alexandrium folks can speak up, but that threshold might not be very high relative to some of the other species. And it's the same for Pseudonychia. If there's, you know, Pseudonychia is weird because it's not always a toxin producer. But if you do have a toxic bloom, sometimes there are not that many cells that we see. And so whether or not we want to call that a bloom, or just be aware that it's there, and you know, again, as Dean said, keep your eyes on things and try to spread that that word and contact somebody else who can, you know, again, kind of confirm things and know whether or not there should be concern there. But yeah, we have, even for Karenia, we have one that's more tied to public health, and then one that's more tied to biomass that we use. And so, and we set those based on our program, and we change them as needed. So it's not a great answer, but it's flexible.
Yeah, right. It seems to be a flexible, and again, probably very much dependent on what's the organism you're talking about, but also, you know, the same number of organisms may not have the same impacts in different areas as well. So it is probably also tied to the local environment and everything else. Yeah, great. Anybody else want to jump on that?
I just want to say one thing. We have Alexandrium blooms here in the Northeast, and in the Gulf of Maine, if we see a concentration of cells of 40,000 cells per liter, that's considered a really dense bloom, a big bloom. But right around the corner from Woods Hole, where I'm stationed, we do work in the Nauset Marsh, and typically we'll see Alexandrium concentrations there, bloom concentrations of hundreds of thousands of cells per liter. So what is a bloom in one area may not necessarily be a bloom in another area. So it's, you know, what you were alluding to, it may be the dominant species in the water sample that you're collecting, or it's relative to the area that you're sampling also.
Yeah, and I would just add to that in the Gulf of Maine, we don't even have to get up into the thousands of cells per liter for toxicity to start occurring in shellfish. You can get up to a couple hundred cells per liter, which may not look like that many at all under a microscope, but is still enough to be causing toxicity.
Right. Yeah. Dean, did you want to jump in?
The concept of bloom has always been difficult for me as a phytoplankton person, because as you see, the various concentrations can have different effects. One thing, like down in Southeast, you cannot see large numbers of Alexandrium in the water column, but you can get high toxicity in the shellfish because you're not seeing those numbers maybe in the surface waters. During the winter, you get these events that will stir up the bottom and resuspend material in which blue mussels or whelks or any of those organisms can filter, and they filter large amounts of water. You're talking, you're talking hundreds of gallons in some cases of some of the shellfish. And so you can see these without really noticing a bloom. And as Kate had mentioned, you can be offshore in Miami and on the Gulf side, and all of a sudden you're getting toxin irritants and you don't really see the large numbers or discolored water. So it's hard to pick up sometimes. So those numbers are variable.
Yeah, yeah, right, great point. Great, I think that.
There's a question in the chat that's asking about unusual behavior in marine mammals, and I'll talk about birds also. If you see a marine mammal that's not acting right, maybe overly aggressive or swimming in circles or spinning, those types of things, you should notice that and report it to somebody. If you see birds on the beach that can't hold their heads up, they have a head droop, that's usually an indication that they have some type of toxin. Or if they're staggering on the beach, acting like they're drunk, it's another indication of toxicity. And so those odd behaviors are the things that I was talking about.
Yep. Yeah, thanks for jumping on that. There's another question in the chat, which is how the species are named, I think. I don't know if anybody knows that. This is a kind of a taxonomist quirk sometimes, but any fun stories anybody has about the specific species of any of these harmful algal blooms?
Okay, I'll chip in on this one. Working in Texas for a while, all of a sudden the water in the lagoons was turning a dark brown, and we were having problems with fish kills and things. And this was related to a brown tide similar to the one they had up in Narragansett. And that organism was named, and the one in Texas, since it was similar, I got to name it, and I called it Oreo, which is a golden umbra because it had a golden halo, and it was found in a lagoon, so it got the specific name Lagunensis. And so you try to give it a taxonomic or a locale type of thing. Or you can name it after somebody like Mikimoto. What?
I was going to say sometimes it's named after a person, and now I know who to thank every time I have to write out Oriumbra.
That's me. Yeah.
It's got a lot of vowels.
Great. Yeah, that's great insight. Gwen asked, when dealing with fixed samples, particularly Lugol's, how do the colors change of these HABs? So yeah, for people that don't know, sometimes taking just a water sample, if you can't keep it frozen, or it's going to be shipped for a long time, or it's, you know, we want to preserve it for longer than a few days, you need to fix it. And one of the most common ways of fixing it is with a solution called Lugol's. And so anybody have experience with Lugol's and how that might change the color of these species?
Yeah, we use Lugol's. Okay,
go ahead, Evie. My answer is going to be very generic, so I'll let you tackle.
I was going to be more specific. So we've used Lugol's to preserve some samples and look for Alexandrium, and I've noticed that those cells end up being a lot darker. So I have to rely more on the shape than that like golden color that is so nice to see in live cultures. Kate, what did you want to say?
I was just going to say Lugol's is one of those fixatives that's really dark. So it makes everything a little bit darker. And you could see in some of the images that we showed today that there is still quite a bit of variability in terms of the pigmentation across the different species. And you still can see that variability with the Lugol's. So the darker cells will get really dark and the not so dark cells will still get darker, but you do typically tend to see that gradation. And you can use sodium thiosulfate to try to complex out the Lugol's. But in terms of how well that preserves all of the different features, you know, in the cells that you might be able to see in a live sample, you're still going to lose some of that after the Lugol's fixation.
Yeah, great.
And I would suggest trying to get a view of the live cells first before it's fixed, or one side by side. And the Lugol's, depending on how heavy-handed you are making the fixation, try to make it a light tea color. You don't want it a nice dark purple, because those cells, they'll all be little purple dots. So
right,
use it sparingly.
Mm-hmm. Yeah, great.
And Thomas, I just wanted the question that Danielle posed about marine mammals and HABs and the potential impacts or unusual behaviors to look for. I just wanted to let folks know I put into the chat a YouTube link to a video that we made with Kathy LaFay out of NOAA Warren West that talks specifically about harmful algal blooms and marine mammals. And there's great information at the very end talking about the Alaska Marine Mammal Stranding Network as well. So feel free to check that out if that's something you guys are curious about.
Great. Okay, another interesting question here that maybe we'll make this the last one before we move on to other quick topics. There's a question here about if there's any heuristic for environmental conditions that are relative rather than absolute. So in other words, if we were to see a change of several degrees Celsius in the water temperature, would we then be more vigilant for a particular organism? And I can throw it to anybody, but I'll just say real quick, I think probably that's true, but there probably is also a minimum water temperature for many of these organisms before. So even if you go from 0 to 5 degrees Celsius, you may not see anything, but if you go from 10 to 15, you would. So it's still 5 degrees, but you're not hitting that minimum temperature threshold. But anybody want to talk about kind of temperature and other conditions that may trigger a bloom or the presence of these HAB organisms?
We can, if we think just about Pseudonychia, in Alaska, we do see temperate species as well as open ocean species, as well as subpolar species, as well as polar species. And so you can see all of those even at the same location at different times. And so, you know, whether or not there's a quick change that would then cause something else to bloom, that quick change may be reflective of a different water mass moving in. And so, you know, it's hard to predict what might happen, but certainly that could bring about a change in composition. And that would be true for, I think, you know, thinking about some of the Alexandrium and how it responds to temperature, and particularly the cysts probably too.
Yeah, Evie, I think one of the fears is that we're finding a lot of these cysts in the Chukchi and Beaufort seas and the sediment, and that with warmer water temperatures, a lot of these cysts may reemerge and create more blooms in the future, right?
Yeah, exactly. I think, you know, as long as the bottom temperature is between 0 and 2 degrees, you're going to get few to no cysts popping out. But once you raise it to 4 or 5 degrees, that gives them a lot of opportunity to germinate. And then when you have the cells in the water, the rate at which they grow and divide is also temperature dependent. So the cooler it is, the slower they grow. And as it gets warmer, they grow faster up to a certain point. But it can also get too warm for them, at which point they'll kind of die off again.
Yeah. Yeah, that's good to I mean, it's not good, but hopefully we don't get to that point where it gets too warm, because we'll have gone through a lot of hell in between that. But yeah, that's good to remember as well. Great.
So Tom,
Yeah.
a quick question on Alexandrium to the Hui people. If there's a massive resuspension of cyst material off the bed, even though the temperatures aren't there for it to exist or bloom, and organisms are eating those, are the cysts themselves toxic?
Great question.
Yes, the cysts are toxic.
And actually, there's someone on this call who's probably muted right now, but I know Courtney Hart has been working really hard to figure out how cysts ingested by shellfish affect their toxicity, and she has some really interesting results to share from that.
Thanks.
Sorry to call you out, Courtney. I mean to...
That's okay. I don't have any answers. I would love to know how toxic a cyst actually is. There doesn't seem to be a lot of modern work showing how relatively toxic it is. I found with gooey ducks that they can consume them, but they don't seem to readily in the winter time any more than anything else. And I found that the couple of clams that I've studied that do have cysts in them weren't necessarily more toxic than their neighbor gooey duck clams that didn't have cysts in their gut. So no definitive answers, but if anyone's looking for a research project, I think finding out how toxic a cyst is in Southeast Alaska or in certain areas would be really interesting to know.
So
Yeah.
I based my comment on cyst toxicity on some laboratory cultured cysts that we've been able to analyze. The interesting thing about Alexandrium cysts is that they have a very resistant coating on the cysts, sporopollenin, and that may prevent them from being digested by shellfish and other organisms. So the toxin may not be actually liberated when shellfish or crab or whatever ingests them. But when we can break them apart in the laboratory via sonication, we do find that they do harbor toxin.
Great question. That's thanks for that. Okay, I think we should probably move on just because we want to make sure we have a little bit of time for the sampling. I just touch on a couple quick more things. Let me just represent this. I'll just do this really quickly. If we don't have any quick answers to this from the panel of experts, we shouldn't spend too much time on it. But we did get a few images from participants that I just wanted to touch on real quick. So This was sent in by Susana Gonzalez that's working down in Juneau right now. And a few quick pictures here. Is there anything that jumps out at any of you as to what these may be in these different slides? This one C here, being a non-expert, looks like a dinophysis, but am I being fooled? I know I'm putting all of you on the spot, but anybody have any input on any of these?
Dave and I were actually looking at this one before the call, and we weren't sure exactly what it is. It looks like its theca might be falling off a little bit. There's that kind of like arc on the side that kind of looks like the Dave called it the wing of the dinophysis. But I don't know, Dave, would you say that looks more like a broken theca?
It's hard to say. It could be a sort of a view from the bottom of the cell. And so maybe that's the crown that you're seeing, and then the wing that you're seeing on the bottom of the cell. That's it. So what Evie and I also were discussing is that you need to look at your whole sample sort of in its entirety, kind of scan it and see what the dominant players are. And then as you're going along trying to make identifications, you can say, oh yeah, well, it kind of has the characteristics of this dinophysis cell, but it's, you know, it's on a weird angle. And Kate mentioned this also, where Karie cells, if they're in the wrong orientation, you're just not gonna see the typical shape. So I think it's important to assess your whole sample and then try to count it, you know, quickly assess it and then try to count it. And hopefully that'll help sort of, you know, give you a better, clearer idea of what you're seeing in your sample.
Yeah. Yeah, I think that's a great point. And so, you know, if you're able to take pictures under the microscope, that's fantastic. And especially if, you know, as we're talking about the orientation, if you're able to take multiple pictures, if they're moving around to see the cells in different orientations, that's very helpful. But this is by no means easy. And I think that one of the takeaways of this workshop is that we need eyes on the water and to see through it, but to, you know, don't feel like you are going to be able to or should be expected to know exactly what you're looking at as soon as you look at it. This is difficult. Experts spend a lot of time, you know, training and learning how to do this. So if you take pictures and feel free to send them to me and I can redirect them or contact experts directly, but take multiple pictures at different angles, and water samples is even better, but obviously harder to ship around. Oh, Kate says one trick that Karen Stedinger taught us: if you save cat whiskers, you can use them to gently move cells. Interesting. I guess you need to have a cat first, but if you have a cat, you can save the cat whiskers to move them around. It's good point. Okay, so yeah.
Or Thomas, I was just going to add, Kate, I love that. Yeah, Karen told us the same thing when I was there. One of the things that I use as well, depending on how you're looking at your samples, is you know, for us, we have our little slide cover on, and you can kind of, if you nudge the edge of the slide cover with a pencil, sometimes they'll roll for you. Obviously, if it's something that's chained up, that might not happen. But for like a single celled, especially dinoflagellate, you might get it to roll, and then taking some photos, especially looking at how the plates are connected, can really help do the identification later on.
Yeah, and all this is obviously, we're talking about, you're already at a microscope, you've already put things on a slide. There's a lot of training that goes to that as well. So, and stuff that's better done in person or in smaller groups. So this wasn't the point of this workshop. This was just, once you're looking at things under the microscope, to give you a little bit of indication of what you might be seeing and help you out there. But obviously, microscope work is another workshop that we may want to put on later, maybe once COVID kind of dies down and we can have more in-person workshops as well, because that's a whole nother thing that is part of this process too.
These were great images to stump the experts. I would suggest, though, if they're going to send an image, at least give us an idea of the magnification they're using. because right here I can see that there were different magnifications used.
Yeah.
And it would be helpful, at least getting a size too.
Yeah, no, that's a great point. Thank you for that. We did get another one from Nicole that I don't think we're going to have time to go through, but she sent us these, and I can circle back with you about these. But she did say that these were all under 40 tons, and so that's definitely going to be helpful. Yeah. So I don't want to take too much more time. So one more thing we just wanted to mention were additional resources for everybody. So obviously, this was just kind of a quick overview. The AHAB website has a few resources and links under this tab here, as well as at the bottom, Ask an Expert, which is a Google form that you fill out that goes to me that I can then redirect, or you can contact me directly. The experts that are here on this panel, and thank you so much for sharing your expertise, are also a resource. Again, I can get you in touch with them. And if you're really into this, if this got you really excited, I'll put in a plug for the Bigelow National Center for Marine Algae and Microbiota course. It takes place in August, and this August one is full, but August 2022, they should have it as well, called Taxonomic Identification of Harmful Algae in US Marine Waters. This is kind of the gold standard. A lot of people on the expert panel have gone through this course. So it's a multiple week course. It's pretty intensive. But that's kind of, if you're really, really excited about this stuff, this is where you would get a lot of that information. And then I think we want to show this as well. So on the left here is the Kachemak Bay National Estuarine Research Reserve phytoplankton guide. That's really great, pretty extensive. Oh, sorry about that. And on the right here is the resource that Dave was holding up there. That's also a really great hard copy resource to have. I don't know if there's a digital version of it, but that's also a really nice one. All right, and if there's nothing else, I just want Rosie. Yep.
Yeah, I also, before we do a quick bio break, just wanted to let folks know who are going to hop off before we get to the sampling section, that we are going to be sharing with you a training evaluation. I just put it in the chat box, and that if you wouldn't mind taking this, there's about like 6 questions on this survey. It will really help Thomas and I kind of get some feedback about what you liked from this training, what you found helpful, whether you're planning on using this information in your work, and will help guide us to see if there are some harmful algal bloom trainings that we can help deliver in the future that you'd be interested in.
Yeah, thank you. Thanks for that. Yeah, please, that would be great. Really helpful for us to fill out that evaluation. I want to thank all the experts again, Rosie, Dave, Evie, Karie, Dean, Kate, thank you so much. This was really, really helpful for me. I'm sure for a lot of people as well. I'll have the recording up of this section, and we're going to stop the recording for the next one. And eventually, I'm not making any promises for a timeline, but I would love to integrate a lot of what was discussed here into a kind of field guide PDF that can be kind of printed and laminated for people to kind of carry around with them if they're going to do a lot of microscopy work on these. But that'll be down the road. But this was already a fantastic start to this overview of the HAB species that we have in Alaska. So thank you all. We'll take a quick break. Anybody who wants to, feel free to jump off. But should we make it 10 minutes still, Rosie, or would you like to come back maybe in 5 minutes?
Let's aim for 5. We were planning on wrapping up today at 10:45, so it gives us about 15 minutes. So let's aim for a 5-minute break, and we'll see if we can quickly go over the sampling protocols. For folks on the sampling protocols that we'll be going over are methodologies that we follow at the reserve, in Southeast Alaska as well, and then also by the Phytoplankton Monitoring Network. So if you're already trained in that, it might not necessarily be super useful for you, but you're more than welcome to stay on and join us. But yeah, let's say aim to take a break, and we'll get going at 10:35.
Perfect. Thank you so much, everybody. Anybody who wants to drop off, thank you so much. Appreciate it. And I will stop the recording.